Hi, my name is RobinCheung, and I will be presenting spatial temporal analysis of the Hawaiian green sea turtle strandings on Maui, Hawaii from 2017 to 2024. So there are 7 species found worldwide, including olive ridley, the Kemp's ridley, the loggerhead, the leatherback, the hawksbill, the green sea turtle, and the flatback. Of those, 5 are found in the Hawaiian waters, which include this green sea turtle, the hawksbill, the leatherback, the loggerhead, and the olive ridley. So Hawaiian green sea turtles are a very unique population due to behaviors that are not traditionally seen within the species of other locations, such as basking on beaches. So it plays a critical ecological role in health and sustainability in marine ecosystems. Also, strandings are a window into the population. They can show population demographics, mortalities, such as mortalities, disease, and regional biodiversity. Also, to better inform conservation efforts by understanding environmental and anthropogenic factors. So where does the data come from? The data comes from the Marine Institute of Maui Ocean Centers, and this is owned, and they work alongside with NOAA Fisheries. They have a call center that reports sea turtle strandings from the public, and then they have trained staff and volunteers that respond to these calls. The data about each stranding is then recorded into a database, and that database is owned by NOAA Fisheries. And then you would need to have permission from NOAA in order to access the database. So research focus. So we're looking at which geographical areas exhibit statistically significant hot or cold spots of the Hawaiian green sea turtle. What are the demographic breakdowns of strandings and what patterns emerge? And what proportional of stranded green sea turtles are found alive versus deceased? And how are they correlated with entanglement type? What spatial distributions can be observed for different stranding causes? And how are they connected to other variables in the data? Now, what seasonal or monthly trends emerge? And are there any repeat strandings, and do they reveal significant patterns or insights? So data was collected from 2017 to 2024. Categorical fields were standardized, and then cases that showed inconsistencies or outliers were excluded from the analysis. Only Hawaiian green sea turtles were evaluated, as these were the most common sea turtles seen around the island of Hawaii. And then that also gave us a total of 1,297 strandings that were used for this project. So GPS coordinates these are the attributes used for analysis, which includes GPS coordinates, regions, age class, sex, condition of the turtle, and then cause of strandings, disease, presence of fishing gear, line ingestion, and pit tag information, which is a type of microchip that can be identified, that can identify each turtle, and then date. So the study area includes just the island of Maui, and we have locations. This map is showing the locations of the Hawaiian green sea turtle strandings within the study area. So this is an overview of the geographical methods and summarizes the key analytical approaches that was used for this study. And then first we're going to look at the average nearest neighbor. This is to analyze the average distance from one green sea turtle to the next, and then determine if the clustered, random, or dispersed is the population. Our results indicate that this is a highly clustered population, and it gives us our observed mean distance at 91.25 meters and our expected mean distance at 762.4 meters. And this will be our starting point for further analysis. We then did the incremental spatial autocorrelation. This determines which spatial scales and clusterings is most pronounced at varying distances. 2 tests were performed, one at 10 and 20 bands, 700 and 750 meter starting points, and then 50 and 25 meter increments. The results showed that there was a clustering that started at 800 meters, and then the maximum at 1,025 meters. So this was our scale that we used for the hotspot analysis as well as our kernel density estimation. So looking at our Ripley's K function, this assesses clustering across multiple spatial scales, giving us a continued view from small to large distances. So the results show us significant clustering began at 2,600 meters, suggesting that our strongest is at the 1,000 to 1,025 meters, but is persistent across a broader distance scale. And then it reinforces our ISA findings and ensures that the spatial weighting and kernel parameters are appropriately scaled. So looking at our kernel density estimation, which gives us a calculation that calculates a smooth and continuous surface showing density layers. Red is our highest densities of sea turtle strandings, and yellow would be our lowest densities. So looking at hotspot analysis, we're identifying statistically significant hot and cold spot strandings. It returns 3 key indicators: our GIZ score, which measures clustering strength. The higher the positive scores is a stronger hotspot, and the negative scores would indicate a stronger cold spot. Our GIP value, which assesses the statistical significance of clustering. And then our GBIN classifies our clustering values. +3 would be 99% confidence of hotspot, and a -3 would be 99% confidence of a cold spot, and 0 would be random distribution. So this is the map that shows the results of my of the hotspot analysis. And for the most part, both the KDE and the hotspot analysis agreed with each other. However, the disciplinary that occurred, there was dispensary that occurred between the north and south of Kihei. And this is just showing that it indicates that using multiple spatial analysis to cross-validate and to better understand underlying spatial process is very highly recommended. All right, so looking at age class distribution. So age class using symbology was grouped into categories based on straight carapace length, and that includes hatchling, juvenile, subadult, adult, and undetermined. Of that, juveniles represented 36.8 of the strandings, and then subadult was 36.1, and adults were 23.1. The last 4 was undetermined and hatchling, and the hatchling was only 1 case, so kind of an outlier. So the hotspot analysis for age class is actually we're looking at the stranding based on age class. So our hotspots is actually clustering of adults and subadults, and then our cold spots are clustering of juveniles. So showing a high concentration on the western and lower south and northern side of the southern part of juveniles, and then the central is mostly adults. So the main 3 stranding causes in this group is entanglement line, foul hooked, and undetermined cause. And then for region, south, which is had our highest strandings of juveniles and subadults, west is second highest of juveniles and subadults, and central was moderate strandings for adults. So live versus deceased strandings. We're looking at percentages with a pie chart showing the percentage of population that is hit, and juveniles is hit the hardest, as you see in the dark blue. Stranding causes for live is considered entanglement line, foul hooked, and hooked mouth. And then the leading stranding cause is undetermined causes and entanglement line. So no further analysis was performed on sex due to the undetermined amount of 77.2%. So looking at stranding cause distribution. So the list of stranding causes are foul hooked, which is where a hook catches onto any part of the turtle's body except for its mouth. Geographic entrapment, line ingestion, predation, undetermined, hooked mouth, entanglement line and net, human take, buoyancy disorder, which is the inability for them to swim due to gas being trapped somewhere in their bodies, impact injury and false alarm. So of these of these 5 strandings, 5 showed significant clustering, minus entanglement line, and that included foul-hooked, geographic entrapment, predation, line ingestion, and undetermined. If we look separately at entanglement line, which this is the most common stranding out of 629 cases, 493 showed no significant clustering. 30 cases did show high entanglement areas, while 106 showed low entanglement areas. So even though they were broadly distributed across Maui, there's definitely areas where there's high-risk areas as well as low-risk areas. So looking at fishing lines and fishing hooks, 2 types of fishing lines are generally used: monofilament, which is a buoyant water absorbent line that has stretching capabilities, and then braided, which is a thicker fishing line with no stretching capabilities. Of those, monofilament had the highest count, followed by braided. Entanglement line was highly associated with monofilament and braided, as well as foul hooked, which came in second, but still associated with monofilament and braided. And then hooked mouth and line adjustment was associated with monofilament. So looking at hook types, no hook had the highest, which is 861, which had entanglement and undetermined. And then undescribed had 214 associated with foul hooked entanglement line and hooked mouth, as well as circle with 155. And then J hooks and other hooks are categorized with foul hooked association. But overall, all hook types were associated with foul hooked strandings, which is represented in the red on the chart. So looking at buoyancy disorder, impact injury, and injury, this map shows a geographical overview of select stranding causes that could potentially influenced by human marine activity. While there's no causal relationships that are implied, that these offer important context and understanding possible events that are occurring. So to further investigate potential clustering analysis, a dip scan analysis was conducted. This identifies identifies significant strandings based on density thresholds, as well as which involved minimum features per cluster at 3, and then the search distance was at 1 kilometer. The results also indicated 2 distinct clusters. However, the majority were considered generally distributed. So by using the buffer tool, a 2-kilometer buffer for a more targeted of the stranding areas that occurred near boating facilities. This captures additional strandings that were outside the DB scan, which can show potential spatial relationships between these human use areas and nearby stranding events. So looking at temporal FP and line ingestion trends. So line ingestion, monofilament, and live sea turtle strandings were peaks in the summer, juveniles peaks in the late fall and winter, and then subadults peaks in midsummer. Entanglement line peaks in spring and summer, and South Coast we see consistent all year long. So FP, which is a disease that is susceptible to green sea turtles, and it's a collar-like tumor that can grow anywhere on their bodies or in their bodies, including their mouth and eyes. 87.3 did not show any signs of FP. 9.6% did show signs, and then unknown was 3.2%. And then stranding caused with line ingestion, 26.6 of the strandings had some sort of line ingestion versus none at 73.3%, and undetermined was at 0.1%. So repeat offenders. So looking at sea turtles with their pit tag information, and then sea turtles that were stranded 3 times or more gave us a total of 22 green sea turtles that were evaluated. So interesting findings within these turtles is that turtle number 4 was recorded to be stranded on the west and south regions. This was unusual, while all the other turtles were recorded within a short distance from each of their strandings. Turtle number 15 was clean of FP for the first two strandings and then became positive on the last stranding. And 18 out of the 22 turtles had ingested fishing line. And the majority of the H class lies within the juvenile subadult. So conclusion. We are looking at stranding events that were more prevalent in the southern and western regions. Age-specific distribution patterns were present, including age strandings in central and southern regions, and juveniles are concentrated in the western regions as well as the southern, northern, and southern parts of the southern region. The most vulnerable were juveniles and subadults. This can have a negative influence on reproductive population and long-term risk to population stability. Entanglement line was the leading cause of strandings, followed by foul hooks. And then monofilament fishing line and circle hooks were recorded as the highest percentage for fishing equipment. So how does this benefit? This can help conservation actions, habitat protection and awareness, public awareness campaigns, beach and reef cleanups, additional research outreach, and direction and development of resources. So there are variables that can influence stranding sites and seasonal anthropogenic issues as well. That includes visitor density. This is a map showing the hotels within Maui. And then also, due to the island's rugged terrain, there are some populated areas that are not observed or observation is limited. So as a result, there's some potential strandings that are being unrecorded simply because the areas are not visited or visually monitored. And then additional data evaluation, such as including benthic and habitat zones, injuries that involve the different age classes, and entanglement shift over time as some examples. And thank you for being here. And if you have any questions, I am now open for questions.